So in this talk today, it will be a lot of experiments and then little bit, there'll be a little bit about theory and a little bit about simulations, but it has nothing to do with GPU. So, we can, those who are interested in discussing GPU, we can do it afterwards, okay? But it's basically on transition from catch to sleep bonds in cell adhesion molecules. So this is a very interesting phenomenon that a while back people have observed in the experiment, and then I've done some work when I was doing a postdoc, and up until now we do some work that is related to this event. Typically, when a force, a mechanical perturbation is applied to the bond becomes weaker, and then it breaks. Strangely enough, in case of adhesion complexes between certain group of proteins, when the force is applied to the bond initially becomes stronger, and only after it passes through some threshold, it becomes weaker, as we expected, and then it breaks. Okay, so this strange behavior has been termed as catch-sleep behavior. Catch basically means catch, and hold to, and sleep means sleep. Sliding, slipping through. So I want to talk about first the experiments when it was observed, and then our work, theoretical work based on that. For that phenomenon. So the outline is, the phenomenon is directly related to leukocyte rolling. So these are the cells, the blood cells in our bloodstream, and they roll on cardiovascular surfaces, and I want to talk about that. And then the catch sleep is directly related to the so-called shear threshold effect, and I will talk about that. And then after I will describe to you this part, I will then go and describe to you the single molecule spectroscopy experiments. So these are the new experiments on single biomolecules or single protein-protein complexes or aggregates. I will describe to you the basics of this method, and then, in particular, one of the realizations is AFM, atomic force microscopy, and then I will talk about the experiment where people first observed catch-sleep response of these bonds to mechanical perturbation or external stress, and then I will describe to you our two-state kinetic model that we came up with that describes the catch-sleep transition and pretty much captures the behavior of cell adhesion complexes subject to pulling force. And then in the third part, if I'll have time, I will talk about simulations of p-selectin forced unbinding. And so the catch-sleep transition has been observed for the family of selectin receptors, and p-selectin is just one of those one of that type of receptors. And then, so in the third part, I'll be talking about coarse-grained models and of proteins and protein-protein complexes, and then the results of our simulations of p-selectin, PSGL1. So this is receptor and this is counter receptor. So we're talking about receptor ligand complex. So these are receptor ligand complexes. The biological, biomolecular complexes. Okay. So typically, let me show a movie. Maybe it will be. I don't. Threshold roll Vivo. Okay, so this is one movie that shows to you that in our blood vessels. So this is the blood flow. Okay, that's a slice. I don't know who made this movie. It's a bad quality movie, but probably 10 years ago or something like that. But what you see is that the leukocytes, I mean the blood cells, they do not fly. I mean, they don't float with blood, but they roll on the surface, just like a bicycle wheel, So it doesn't flow. Strangely enough, it doesn't flow, but it sticks to the surface and then it rolls on the surface like a bicycle wheel. So there are many cells. So there are leukocytes, C lymphocytes, neutrophils, platelets. There are many cells in the blood, and almost all of them have selectin receptors expressed on their surface. Okay, so that's why. So these are although these are neutrophils, if you were to look at leukocytes, you will find the same picture. They'll be rolling on the surface. Okay, so now this is the in vivo experiments, and then one can also do in vitro experiments. And so this is the movie of rolling in vitro. So basically, these are the now these are the neutrophils. Okay, so these are the neutrophils cells. So they have been extracted from blood, and then they're put in the chamber, and then there's a flow of liquid. Okay, and you see that some of them are flying by. Some of them, you see these, bullets flying by. These are the flying because it's not in vivo experiment; it's in vitro experiment. But most of them are rolling. Okay, these guys are rolling. So, and you see that you can parameterize. You can do science on this by looking at the average velocity. Okay, you can look at, then you can zoom. You can say what is the tether frequency? How frequent do they tether? They fly and then they stop. So that's a tether, and then they roll. Okay, and then they detach from the surface, and again they fly for some time. Again they tether. So you can talk about the tether frequency. You can talk about the waiting time. So, for instance, sometimes it moves in the stop and go manner. So it goes and then it stops, then it waits, then again it goes, then it stops, then it waits again. So you can discuss the distribution of waiting times. So there are many levels of description of this interesting phenomena if one is using statistics and probability theory. Okay. So, but these are basically the experiments, and in this particular experiment, you can see this as just a camera. So people brought this camera, and then they have this flow chamber, and then they have P-selectins. So in this experiment, how it was done, you have these P-selectin receptors on the surface, and then you have these cells. They are rolling around, right? They're rolling over, and in this direction. So the purified P-selectins are on the surface, and then neutrophils or any other blood cells are in the flow. Okay, you can do it. So the interesting thing, what scientists have found, was very counterintuitive, and it has been termed as shear threshold effect. What exactly is shear threshold effect? Look. So this is the shear stress, 1.5 dynes per square meter. Okay, now 0.2 you decrease, it moves faster. You increase, it moves slower. So when you increase the shear stress, they have to move faster, but they move slower. You decrease, so you see, you increase, it moves slower. Then you decrease, it moves faster. So now you increase, it has to move even faster, but yet it moves slower. You see? It's very counterintuitive, and people did not understand. So they just termed it, shear threshold effect. Okay? So this is very interesting. And then like I said, this is a cartoon of a blood flow. Okay, so if we were to go back to in vivo description, then these are the blood cells, and this is the endothelium. So these are the surface of our blood vessels. So on that endothelium, we have P-selectin receptors, okay, expressed. So these are endothelial cells, and there's some P-selectin receptors expressed on the surface, okay. And these blood cells, they have counter receptors. PSGL1 is one of them. That's the pair for P-selectin. So PSGL1 is expressed on the surface of blood cells, and P-selectin receptors are expressed on the surface of blood vessel walls. Okay,